Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. First, I would like to thank to ISFAP 2021 organizer for giving me opportunity to present in this conference. My name is Isnaeni from Research Center for Physics, International Research and Innovation Agency, BRIN. My talk is about two-photon emission of fluorescent carbon dot made of urea and trisodium citrate. My co-authors are Ion Sugiarto and Wildan Panjitrisna. I will start my talk with introduction, followed by research purpose, method, result, and discussion. I will close my talk with a summary. Many materials have fluorescent properties, especially nanoparticles such as nanodiamonds, metallic nanoparticle, nonlinear crystal, and quantum dots. Luminescent quantum dot has been studied by many researchers for decades. There are several types of quantum dots, such as semiconductor quantum dot, oxide quantum dot, and organic quantum dot, including graphene quantum dot and carbon quantum dot. We will focus on this work on carbon quantum dot. Carbon quantum dot can emit a certain light upon an excitation light. Two main mechanisms of photoluminescence are down conversion luminescence, also known as one-photon emission, and up conversion luminescence, which is also known as a two-photon emission. Many researchers have tried to use quantum dot to detect location of cancer. Others have to try to utilize quantum dot for photodynamic therapy to cure cancer. The utilization of quantum dot for biomedical application is due to small size of quantum dot around 10 nanometers. In biomedical application, the photoluminescence is the most common detection technique. Quantum dots that have attached to a specific cell or antibody are then excited using proper light. The emission will give information on the disease and treatment. Using down conversion mechanism, UV or blue laser is used. However, UV and blue laser are dangerous for living tissue. Meanwhile, two-photon emission or up conversion photoluminescence use red or infrared light which have low photon energy that is harmless to living tissue. However, utilizing quantum dot in the biomedical process is still facing several challenges. To be safely applied in living bodies, non-toxic quantum dot must be used. In this case, carbon quantum dot is one of the safest quantum dots nowadays. Another challenge is the potency of two-photon emission in the biomedical. There are only few research on investigating the photoluminescence of carbon quantum dot under two-photon emission mechanism. However, many works can be explored regarding the two-photon emission of carbon quantum dots. This becomes the motivation for us to conduct this research. The purpose of this work are, number one, to synthesize carbon quantum dots that are made of urea and trisodium citrate. Number two, to observe the possibility of two-photon emission of synthesized carbon quantum dot. And number three, to compare down conversion photoluminescence and two-photon emission mechanism. We limited our work to only one type of carbon quantum dot, and we only focus on the optical property of carbon quantum dot. We hope that our result will bring more information of the possibility of carbon quantum dot in medical application. Carbon quantum dot were synthesized using a simple microwave technique. Trisodium citrate and urea were dissolved in distilled water and stirred in a beaker glass. The mixture solution was then heated in the domestic microwave for about 10 minutes until we got dark brown flake. The flake was then crushed using mortar until we got fine powder. To make carbon quantum dot solution, we dissolve 1 milligram of the fine powder with 50 milliliter distilled water. The solution was then filtered before optical measurement. Photoluminescence of carbon quantum dot was done using a simple setup as shown here. A laser source was focused using a lens onto a carbon quantum dot in a quartz cuvette. The emission from carbon quantum dot was then directed into a spectrophotometer. For down conversion photoluminescence, we used three different laser sources: 405 nanometer, 420 nanometer, 450 nanometer diode lasers. For two-photon emission, we use femtosecond laser from MITAI with 800 nanometer, 850 nanometer, and 900 nanometer wavelength. In addition, we also measure absorbance of samples. Synthesized carbon quantum dots have a light brown color appearance in liquid form. We noticed that the solution was stable for weeks. Furthermore, the emission of carbon quantum dot was observed under down conversion photoluminescence, figure B, and two-photon emission, figure C. In brief, cyan until green color emission was observed under down conversion photoluminescence. Furthermore, green yellowish color emission was observed under two-photon emission mechanism. The absorption of colloidal carbon quantum dot is shown in this slide. We notice two distinctive absorption peaks of carbon quantum dot. The first peak was about 232 nanometer equals to 5.34 electron volt. The second peak was at 328 nanometer equal to 3.78 electron volt. The first peak was related to the energy transition from pi to pi star state. Meanwhile, the second peak was related to the energy transition from n to pi star state, which is originated from the surface functional growth of carbon quantum dot. These figures are the emission spectra of the carbon quantum dot excited using different lasers. The emission color of carbon quantum dot by 405 nanometer laser is blue-green color. The emission color shifts to the green color when carbon quantum dot were excited by 450 nanometer laser. This phenomena is known as red shift. Based on the absorption spectra, we noticed that after 400 nanometer, the absorption curve tends to flatten. This means no absorption process. However, laser 405, 432, and 450 nanometer were able to excite the carbon quantum dot, resulting in a good emission spectra. This indicates that there is still an absorption process by carbon quantum dot for wavelength longer than 400 nanometers. Furthermore, using two-photon emission mechanism can give nice emission spectra shown in the right figure. Longer excitation wavelength made lower intensity of the emission. We notice green-yellowish color emission from the cuvette excited by femtolation laser. Although the femtosecond laser that we use have a high intensity laser, however, the photon energy was very low. 800 nanometers is only have a 1.55 electron volt energy. Emission spectra obtained by femtosecond laser excited was the proof of the two-photon emission mechanism occurred in the carbon quantum dot. To give detailed information, we plotted the emission peak wavelength of each emission spectra as shown in this slide. We can see from that figure under down conversion photoluminescence, the emission peak wavelength of carbon quantum dot noted by blue diamond. Meanwhile, under two-photon emission, the carbon dot peak wavelength is noted by a red star in this figure. Actually, in this work, we selected 6 laser excitation on purpose. 800 nanometer laser from femtosecond laser is approximately half photon energy as the 405 nanometer laser. The different mechanism of one photon and two photon emission is shown in this slide. We generated a simple energy band diagram using data from absorbance and photoluminescence. In down conversion photoluminescence, one photon from a blue laser can excite an electron to 3.06 electron volt above the P start state. This energy can excite an electron to a certain N state level. From this state, the excited electron went to a relaxation state below it. The recombination process was then taking place by emitting lower energy photon as known as the emission color of photoluminescence. Two-photon emission of or upconversion photoluminescence is one type of nonlinear optical mechanism. The most common mechanism is the presence of the so-called virtual state. The photon energy from one photon of 800 nanometer wavelength was only 1.55 electron volt. This energy only excites an electron to a virtual state. Then the second photon came and excited this electron again to a higher energy level. We expected the same relaxation and recombination process happen after this two-photon excitation. Finally, we have successfully synthesized carbon quantum dot made of urea and trisodium citrate. We have shown that our carbon quantum dot were able to give fluorescence under both down conversion and two-photon emission photoluminescence. Using a band diagram, the different mechanism of down and up conversion can be explained really nice. This result may give a good hint for real biomedical application. Thank you very much for your attention.